So now that we've seen how amphibian kidneys work, we're prepared for a deep dive into the structure and function of the mammalian kidney. This is a topic that we're going to approach in stages as we explore the nephron anatomy and then trace the fates of 3 different groups of components. After the anatomy, we're going to look at what happens to the inorganic ions in water, which will include an introduction to one more type of exchange system, the countercurrent multiplier. Once that's covered, we'll follow the fate of urea to see how that gets concentrated. And then from there, we'll look at how other solute reabsorption happens in the proximal convoluted tubule. And then lastly, we're going to look at how blood flow happens through the kidney. Beyond this, we're going to conclude by briefly talking about excretion in insects as another comparison case, and then we're going to touch on some interesting details about the diversity and how nitrogenous wastes are dealt with and excreted by different types of animals. Okay, so for now, the anatomy. So just as with the amphibian nephron, all these structures are usually coiled up, as you can see illustrated here in this image from your textbook. But for the sake of study, we're going to work with the uncoiled version pictured here on the left. To start out with, we need to think about how nephrons are situated relative to the overall shape of the kidney. So as a reminder, here up top is the cortex. That's that outermost portion of the kidney. And then the outer portion of the medulla here. And then the inner portion of the medulla down here. And as you're going to discover really soon, this spatial organization really matters. Okay, a couple other components. So here again is Bowman's capsule. So there can be some variation in exactly where a given Bowman's capsule is situated, but in general, the Bowman's capsules are in the cortex, as you see here. Once again, there's going to be a proximal convoluted tubule and a distal convoluted tubule here and here. But there are a couple of important changes from the amphibian nephron. The first is what's happened to the intermediate segment. In the nephron that's here on the left, this section is dramatically elongated into what's called the loop of Henle. And note that this isn't universally true. In this nephron illustrated over here on the right, which is located further out into the cortex, this nephron only has a short loop and a very small, thin segment. So what I want you to realize is that there can be variation within a kidney with respect to individual nephron function, in addition to there being variation between kidneys of different species. And also note that there are both thick and thin components to the loop of Henle. So here's thick, here's thin. And so this is important because each of these components has slightly different functions associated with that morphological difference. And I also want to point out something that's not illustrated in this image, but the thick portion of the ascending part of the loop of Henle, so up here, that's actually very closely associated with Bowman's capsule. I'm mostly pointing this out in the event that you go on to study renal physiology in greater detail, because this portion of the distal convoluted tubule is involved in aspects of fluid and ion regulation. Along with all of this, I also want you to note how blood vessels are positioned. So here they've got equally long projections into the medulla, and even the shorter nephron has this type of orientation with arteries further out towards the cortex and then veins located deeper in towards the medulla. Okay, and then lastly, the structure that's over here on the left, the collecting duct, is also going to factor very much into the events that we're going to go through. Okay, so that's the general organization. So now we're going to talk about the fates of different types of solutes, starting with the water and inorganic ions. First, I want to show you an illustration of a nephron that I drew for you. And the whole idea here is that you can print out as many copies as you'd like and write directly on them. I find that to be really useful for practice and for keeping track of different aspects of kidney function. Okay, so as with the amphibian nephron, we have the ultrafiltration and production of primary urine at Bowman's capsule. The question for us to answer here is, again, as with the amphibian nephron, how do water and solutes get harvested back out of the primary urine? So first off, things in the proximal convoluted tubule are very much like the amphibian proximal convoluted tubule here. So there's active reabsorption of water or I should say that active reabsorption leads to the reabsorption of water, right? Since water itself doesn't get actively reabsorbed, but also of dissolved solutes. And this uses the same type of active transporters as we've seen before. So, for example, there are some sodium-potassium pumps and secondary active transporters to transport molecules like glucose, plus constitutive aquaporins that allow for this water reabsorption. Along with that, the proximal convoluted tubule also contains urea transport proteins. And so note that the urea transport proteins, they actually function very much like a channel protein would. So they don't engage in any type of active transport, but they do allow diffusion to happen, facilitated diffusion. So as other solutes get transported out, conditions can favor the movement of some urea into the proximal convoluted tubule. And we'll talk more about that later, as I mentioned before. So in any case, as we sketch out what happens next, it becomes important to know that some of the key factors involved here have to do with the ion and osmotic composition of the fluids that are outside of these nephron structures. So in other words, in the interstitial fluids here. And it's also important to know that there are hard upper limits to how much of an osmotic gradient sodium transporters can achieve across a given distance. So by the time that the primary urine reaches the upper portion of the loop of Henle here, it's going to be more or less still isosmotic to blood plasma. So the osmolarity here is going to be right around 300 milliosmoles. The cool part is what happens next in the loop of Henle. What happens is a process called the single effect, which along the loop of Henle can lead to what's known as countercurrent multiplication. So this is different from what we've observed previously with the countercurrent exchange systems, and therefore we need to work through how countercurrent multiplication works. So you're going to have to roll up your sleeves for a bit here. And what it depends on is the spatial organization of transport proteins, as well as the movement of fluid through the two tubes that are parallel to each other, but in opposite directions. So to work our way through this, we're going to start out thinking about these two tubes parallel to each other, okay? So here's tube 1, here's tube 2. Okay, so in our starting conditions, we're going to think about everything being in equilibrium. Okay, so we've got solutes here, solutes here, solutes here. Okay, and these are all aqueous. However, the tube on the left is permeable to water and solutes. Okay? So those can move via passive transport. And in contrast, this tube on the right is impermeable to water. So we can maybe illustrate that by making the walls thicker here. Okay, and it also contains proteins that permit active transport of sodium and chloride out of the tube. Okay. And as a side note, the transport protein arrangement and mechanism in this right-hand tube resemble the marine teleost chloride cells, just oriented in the opposite direction relative to the fluids, so to harvest sodium and chloride out of the fluid inside of this tube. And I really hope that that point actually kind of blows your mind a little right here. Okay, so if sodium and chloride are getting actively transported out of this tube, what that means is that solute concentrations in this interstitial fluid are going to increase, right? Okay. Okay, and that in turn is going to cause diffusion of water out of the left tube, right? So water is going to start diffusing out. And there will be some diffusion of some sodium and chloride into the left-hand tube. Okay, so our end result will be that we're going to have higher solute concentrations in the left tube and interstitial fluid, and removal of water from the left tube to the interstitial fluid. And there's also going to be a decrease in the solute concentrations on the right. Okay, so this in a nutshell is the single effect. Okay, so now we're going to map this back onto the loops of Henle. This is actually exactly the arrangement that we have in the loops of Henle. So in our descending loop of Henle, we have a descending limb that is permeable to water. And then the thick portion of the ascending limb contains transport proteins that will cause active transport of sodium and chloride out. And so again, those are the NKCC transport proteins plus all of the necessary accessory proteins to make that happen. So now we need to look at the overall consequences of that single effect I just showed you along the length of the loop of Henle. And then at the end of this section, I'm going to emphasize why this is so important for allowing mammals to produce concentrated urine. So next we're going to look at a diagram that's often used for illustrating what you get when there's a single effect along a tube with a hairpin turn. So we're going to start out with our solute osmolarity that I mentioned at the end of the proximal convoluted tubule. So that was 300 milliosmoles. So we're going to, in this case, everything in this environment starts out at 300 milliosmoles. So we're now going to imagine that there's active salt transport out of the ascending limb, and then there's going to be water movement out of the descending limb that happens as and this all happens as follows, right? So if we have active transport out, we're going to wind up with an osmolarity inside of the ascending limb here of 200 milliosmoles, and that's going to cause an osmolarity in the interstitial fluid of 400 milliosmoles, which means that this solution, which had been at 300 milliosmoles, there's going to be water that leaves and an equilibration of solutes such that then we wind up with a concentration inside of this descending tube that's also at 400 milliosmoles. Okay, so that's what we have so far. So next we're going to move the fluid in this tube forward by one increment. So we're going to send more concentrated urine here down the descending limb, and we're going to send this more dilute urine up and out of the ascending limb. And then we're going to bring in more fresh primary urine at 300 milliosmoles here at the top of the descending limb. and then this 200 milliosmol urine will leave. Okay. And we've got more 400 milliosmol urine here towards the bottom of the tube and starting to loop back around. Okay, so that's pictured right here. So here's 300 milliosmoles. Here's our 400 milliosmoles. And then here's our 200 milliosmoles on its way out. Okay, so now if our sodium chloride transporters do their job again, remember they're all the way along this length here, we're going to move out another increment of sodium all the way along here. But because this fluid here now is lower in concentration than it was previously, we're going to wind up with an even lower concentration here than before. So here that's pictured as being 150 milliosmoles. We aren't going to be able to establish as extreme of a concentration gradient up here. So now what we've got in the interstitial fluid up here is 350 milliosmoles. And then in the descending limb, we've got 350 milliosmoles once that equilibrates. Okay, so now let's contrast that with what happens down here towards the bottom. So down here, we have some of that 400 milliosmol fluid that started to make it around the bend and back up again. And so we're going to have a steeper change down here, and we're actually going to be able to establish an even higher concentration down here in the interstitial fluid than we were in round one, right? So here we were at 400 milliosmoles. Here, because we're transferring out of a 400 milliosmol concentration, we can now get up to 500 milliosmoles in the interstitial fluid, and also 500 milliosmoles in the descending tube, and we're going to wind up at 300 milliosmoles over here in the ascending tube. And this is going to be a really important point. Okay, so now we're going to increment again. So we're going to have more fluid come in at 300 milliosmoles, and then we're going to have more fluid leave, some of this 150 milliosmoles. And so we also now notice have an even higher osmolarity coming in and around the bottom of the tube, right? We went from 400 here now to 500 here, okay? And then once again, we're still going to have active transport all along the length of this tube, right? So from 150 here now, so if we actively transport out, so now we're going to wind up with an osmolarity in this ascending limb at 125. Out here in the interstitial fluids, we've got 325. We've got our 300 milliosmol coming in, so this is going to equilibrate up at the top of the tube at 325 milliosmoles. And now, by this point, what I'm hoping you can start to see is that as this whole process continues, the net outcome is going to be that the interstitial osmolarity towards the bottom of the tube is much higher than it would have been in the simpler case of just the single effect by itself up here. Okay, so altogether, the combination of the single effect plus countercurrent multiplication is going to result in four things, okay? Okay, so first, it's going to establish an osmotic gradient from the outer portion of the kidney, so from the cortex, towards the inner portion of the kidney, the medulla, right, towards the tip of the loop of Henle, right? So there's cortex, here's medulla. So altogether, in humans, for example, we can get up to an osmolarity at the tip of the medulla of 1200 milliosmoles instead. Okay, the next thing is that this process is going to remove a lot of water from the urine. In that descending limb, right over here. So that the fluid volume that leaves the ascending limb of the loop of Henle is a much smaller volume than the fluid volume that enters the descending limb. And in fact, so if we come back to this, by the time the fluid reaches the collecting duct, so up here, we're actually at less than 5% of the original primary urine volume. That was filtered in way back here at the glomerulus. And then the other thing that's going to happen at this stage is that this whole process is going to harvest back most of the salt, right? Because, again, if we come back to this picture here, right, the osmolarity that's over here leaving the ascending loop of Henle is much lower as the urine heads towards the distal convoluted tubule out of the loop of Henle. And then the last thing I want to emphasize here is that the longer the loops of Henle in an animal, the bigger the gradient that can be established. And so the more water and salts that can be extracted back out again. Okay, so now the last thing we need to do is we need to bring the collecting duct back into the picture. Okay, so here we need to again consider two contrasting situations of diuresis and antidiuresis. So diuresis first because it's simplest. So, and just let me note again, so here what we have leaving the ascending limb is going to be somewhere around maybe 125 milliosmoles, right? So we went from 300 to a departure at 125, and down here we can get up to 1200 milliosmoles thanks to the single effect and countercurrent exchange, right? Okay, so now during diuresis, the collecting duct is impermeable to water. Okay, and so the dilute urine that's up here at the distal convoluted tubule can basically just flow through the collecting duct and out. And there are actually even more sodium transport proteins along this collecting duct. So we can get down to an osmolarity as low as 65 milliosmoles by the time the urine actually leaves from the collecting duct. So that's pretty good for an animal that's seeking to get rid of extra water. All right, so what about antidiuresis? So what I want you to do for a second here is think about what happens if we stick aquaporins in the collecting duct. Right? Okay, so what happens if we stick aquaporins in this collecting duct? Okay? So remember that the osmotic gradient down here is a lot higher than it was when the urine entered the proximal convoluted tubule up here, right? So the consequence is that if this collecting duct is permeable to water, we can start to extract water back out, right? If this is up here is at 125 milliosmoles, so there's a strong osmotic gradient to collect water back out along this collecting duct. And to the point where the urine osmolarity is going to be equal to the osmolarity of the interstitial fluid here, right? So we can get back up to 1200 milliosmoles. Okay, and again, if we have an animal that has even longer loops of Henle, this process can be extended and there's a potential to produce even more concentrated urine. Okay, so this is a lot to make sense of. So we're going to stop here for right now and we're going to pick back up again with a story for urea, where the picture is different from what I've just shown you here for water and salts.